and welcome to our program. Dear students, today we are going to discuss one of the basic topics of developmental biology, that is gametogenesis and fertilization, structure and production of gametes, and finally zygote formation. Let's see today's main objectives. Understanding the modes of germ cell specification, migration of primordial germ cells in developing gonads, particularly in Drosophila, general account of gametogenesis, spermatogenesis and oogenesis, and finally mechanism of fertilization process. Dear friends, as you know that gametogenesis is the process of production of gametes that are formed from the primordial germ cells present in the gonads. The gametes later fuse to each other, a process called fertilization, which leads to development of zygote from which new individuals are formed. This is how life continues. The germ cell from which gametes are formed is the essence of this continuity of life. Under the heading of germ cell, we will focus on different theories related to germ cell and germ cell determination in different animals. Let's discuss first germplasm. Germplasm, which is also known as polar plasm, is a zone found in the cytoplasm of the egg cells of some organisms which contain determinants that will give rise to the germ cell lineage from which gametes are formed. Gametes are sexually dimorphic, that is, male and female gametes. They fuse and form zygote. As the zygote undergoes mitotic divisions, the germplasm is ultimately restricted to a few cells of the embryo. These germ cells then migrate to the gonads. In many animals like vertebrates and invertebrates, the germline, which is a population of cells that differentiates into the process of reproduction, may pass their genetic material to the next progeny, developed separately from the somatic line. These somatic cells are called precursor cells of primordial germ cell. Different animals have different germ cells. In case of nematodes and flies, precursor germ cells are specified autonomously by the cytoplasmic determinants in the egg. These cytoplasmic components of the egg, which are preformed, later become part of specific cell, that's germ cell, during cleavage and is known as germplasm. In animals like salamander, germline specified is conditional and the germ cells are specified by interaction among the neighboring cells by cell-to-cell signaling. This is called induction or epigenesis of germ cell. There is not any cytoplasmic determinant present in the cell of such animals. Now let's discuss the mechanism of primordial germ cell specification. First of all, germplasm theory of heredity. This theory was published in the year 1893 by August Weismann based on the concept of germplasm. This theory was based on four concepts, that's biophore, determinant, its and oidents. However, this theory is not valid now. Second, germ cell determination. It's also called Bovary manifestation for germplasm theory, studied on roundworm Por Ascaris aquarium. Bovary experimentally provided the determinant of germplasm. He described the equatorial and reductional division of the germ cell to form the egg. The first division plane of this roundworm zygote is equatorial, separating the animal and vegetal poles, which can be distinguished based on yolk granules. In Por Ascaris aquarium's blastomere, the chromosome ends disappear and only central part remains intact. This peculiarity is called chromosome diminution. In vegetal blastomere, chromosomes remain normal. During second division, the cell splits meridionally while the vegetal cell again divides equatorially. Initially, both vegetally derived cells have normal chromosomes, but chromosomes of animal pole eventually undergo diminution before third cleavage. Likewise, at 16 cell stage, only two blastomeres have intact chromosomes. One of them eventually undergo chromosomal diminution and gives rise to more somatic cells, while other give rise to germ cells. Thus, the chromosomes are kept intact only in those cells that are destined to become the germ cell. Now we'll go through how germ cell determination takes place in different animals. First, we'll take C. elegans. In C. elegans, a ribonucleoprotein complex termed as P granule, which is luminescent in nature, is found which specifies the germ cell. After fertilization, the randomly scattered P granules localize at the posterior end of the zygote and entire cells are formed from the posterior cytoplasm P1. On subsequent divisions, P granules pass from P1 to P2 cell, then P3 and lastly reside in P4 cell, whose progeny eventually gives rise to the gametes in adult. Dear students, now let's discuss germ cell determination in Drosophila. In Drosophila, at 9th cellular division, the progenital germ cells form a group of pole cells at the posterior region of developing embryo. the nuclei migrate to the posterior region and get surrounded by the pole plasm that's composed of mitochondria, fibrils, and polar granules. The granules contain specific RNA and proteins which play an essential role in the germ cell formation. The next is germ cell determination in zebrafish vertebrate. The germ plasm of zebrafish is composed of granules, mitochondria, and mRNA that are localized in the cleavage furrow of the early dividing egg. At late cleavage, the germ plasm gets localized in 4 cells that subsequently divide and give rise to 4 clusters of progenital germ cell. Dear students, now let's have a brief look at defining feature of germ cells. A germ cell line differentiates from somatic cell line in the following ways. First, germ-line soma interaction. It is important for germ cells to regulate their proliferation, that's maintenance, differentiation, that's division into the sperm and ova, and protection of germ cell genome. These cannot differentiate on their own, and for all these processes to occur, germ cell soma interaction is important. Second, transcription repression. It's also known as inert genome hypothesis. The germ cell generally set apart from soma cell, that's body cell, by transcription repression that prevents transcription of gene into mRNA. Third, conserved DNA regulator. There are some RNAs which regulate the maintenance of the germ-line character in the germ cells and prevent them from entering the somatic differentiation. Our next topic is migration of germ cells. In Drosophila, progenitor germ cell has to move from posterior pole to gonads that are developing from abdomen of the embryo. During early stage of embryogenesis in Drosophila, the hind end moves towards the dorsal side and the germ cell is carried inside the embryo, passively adhered to the moving layer of somatic cell. This will give rise to the midgut, and process is called germ band extension. Next, the germ cells actively move by diapedesis through midgut epithelium, then migrate to the visceral mesoderm and split into 2 groups. Each group associates with one of the somatic gonads developing on either side of the embryo. Dear students, now let's move on to the main topic, gametogenesis. The process of formation of haploid gametes from the undifferentiated diploid germ cells in the gonads for sexual reproduction is called gametogenesis. It's of 2 types, spermatogenesis, which is the formation of male gametes, that's spermatozoa, and oogenesis, which is the formation of female gametes, that's ova. Both involve 3 important phases: multiplication phase, in which the germ cells of the gonads multiply by mitosis; second, growth phase, in which the germ cells grow in size; and lastly, maturation phase, in which the meiosis takes place to produce the gametes. Let's discuss spermatogenesis. In humans, spermatogenesis takes about 65 to 75 days. The seminiferous tubules are lined with immature cells called spermatogonia. These cells develop from primordial germ cells that arise from yolk sac endoderm and enter the testes early in development. Spermatogenesis is a continuous process, but for convenience, it's often divided into 2 states: spermatogenesis and formation of spermatozoa from spermatids, which is known as spermiogenesis. Let's discuss first spermatogenesis. This process has been divided into 3 phases: multiplication phase, growth phase, and maturation phase. First, let's discuss multiplication phase of spermatozoa. The primordial germ cells differentiate into spermatogonia but remain dormant until they begin to undergo mitotic proliferation at puberty. Spermatogonia contain the diploid chromosome number. When these stem cells undergo mitosis, some of the daughter cells remain undifferentiated and serve as reservoir of stem cells. Such cells remain near the basement membrane. The rest of the daughter cells lose contact with the basement membrane of the seminiferous tubule, undergo certain developmental chains, and differentiate into primary spermatocytes. Primary spermatocytes, like spermatogonia, are diploid, that is, they have 46 chromosomes in case of humans. Second phase is growth phase. On sexual maturity of the animal, some spermatogonia stop dividing and grow in size by accumulating cytoplasm and replicating DNA. These cells are now called primary spermatocytes. Each primary spermatocyte enlarges about twice the size of spermatogonia before dividing. Other spermatogonia continue to divide mitotically and produce more spermatogonia for later spermatogenesis. The next phase is maturation phase. It's the division phase in which the spermatids are formed from the primary spermatocyte. the spermatogonia present inside the wall of seminiferous tubules multiply by mitotic division and their number increases and subsequently form primary spermatogonia. In later phase, primary spermatogonia undergo meiosis I, leading to the production of two spermatocytes which have haploid number of chromosomes called secondary spermatocytes. Then it undergoes second meiotic division, which is equivalent to mitosis, leading to formation of 4 haploid spermatids. The next phase in spermatogenesis is spermiogenesis. The final stage of spermatogenesis, called spermiogenesis, involves the maturation of spermatids into spermatozoa. Each spermatid develops a head with an acrosome, which is enzyme-containing granule, and a flagellum, which is a tail. Since there is no cell division in spermiogenesis, each spermatid develops into a single spermatozoon or sperm cell. The release of spermatozoon is known as spermiation. Sperms enter the lumen of the seminiferous tubules and migrate to the ductus epididymis. There, they complete their maturation in 10 to 14 days and become capable of fertilizing an ovum. Spermatozoa are also stored in the vas deferens. Here, they can retain their fertility for up to several months. Dear students, now let's discuss the hormonal control of spermatogenesis. After attaining puberty, there is secretion of gonadotropin-releasing hormone secreted from the hypothalamus. The increasing level of gonadotropin-releasing hormone then acts at pituitary gland and stimulates two gonadotropin hormones, that's luteinizing hormone and follicle-stimulating hormone. Luteinizing hormone acts on the Leydig cells and secrete androgens, which in turn stimulate spermatogenesis. Follicle-stimulating hormone stimulates androgen-binding protein by Sertoli cells. The androgen-binding protein is essential to concentrate testosterone level high enough to initiate and maintain spermatogenesis. The release of sperm from seminiferous tubules is transported by accessory gland. The secretion of accessory gland and sperm constitute the semen. Dear students, now let's move on to the oogenesis. It's the process of formation of reproductive cells in the ovaries. Whereas spermatogenesis begins in male at puberty, oogenesis begins in females before they are even born. It starts in 25-week-old fetus. Egg formation starts before birth but is completed only after fertilization. Dear students, now let's discuss the process of oogenesis in detail. Oogenesis occurs in essentially the same manner as spermatogenesis, with meiosis taking place and resulting germ cells undergoing maturation. Oogenesis also consists of 3 phases: multiplication phase, growth phase, and maturation phase. First, multiplication phase. During fetal development, the germ cells differentiate and undergo mitotic divisions, producing undifferentiated germ cells called oogonia or egg mother cells within the ovaries. Oogonia are diploid stem cells that divide mitotically to produce millions of germ cells and project into the stroma as a cord, the egg tube, which becomes a round mass, the egg nest. Most of these cells degenerate in a process known as artesia. A few, however, develop into larger cells called primary oocytes. It's the future ovum that enters prophase of meiosis I during fetal development but doesn't complete that phase until after puberty. Other oogonia lose the potential to become primary oocytes and form a regular layer, the follicular cells, and surround the primary oocytes to protect and nourish them. The structure thus formed is known as primary follicle. Each primary oocyte is surrounded first by one layer of follicular cells and then by 6 to 7 layers of cuboidal cells called granulosa cells. As the follicle grows, it forms a clear glycoprotein layer called zona pellucida in between the primary oocyte and the granulosa cell. The innermost layer of granulosa cells become firmly attached to the zona pellucida known as corona radiata. The granulose cells gradually begin to secrete follicular fluid, which builds up a cavity called antrum in the center of the follicle, which is now termed as secondary follicle. Next phase is growth phase. The growth phase of the primary oocyte is very long, extending over many years. During this phase, the cytoplasm increases and nucleus enlarges after puberty. The next phase is maturation phase. Each month after puberty, gonadotropins secreted by the anterior pituitary stimulate resumption of oogenesis. Meiosis first resumes in several secondary follicles. The diploid primary oocyte completes meiosis I, producing 2 haploid cells of unequal size, both with 23 chromosomes each in case of humans. In humans, the first meiotic division is reductional. The smaller cell produced by meiosis I is called the first polar body or polocyte, and the larger cell which receives most of the cytoplasm is known as the secondary oocyte. Once the secondary oocyte is formed, it begins meiosis II but is then arrested in metaphase II. The follicle in which these events are taking place is the mature or Graafian follicle, soon to rupture and release its secondary oocyte, which is known as ovulation. The secondary oocyte is then expelled into the uterine tube with the first polar body and corona radiata. If fertilization does not occur, the cells degenerate. However, if sperms are present in the fallopian tube, one penetrates the secondary oocyte and meiosis II resumes. The secondary oocyte divides into two haploid cells, again of unequal size. The larger is the ootid, and a very small cell that is also produced is the secondary polar body. The first polar body may divide at about the same time and produce two polar bodies which also degenerate. Thus, one primary oocyte gives rise to a single gamete and ovum. By contrast, in males, one primary spermatocyte gives rise to 4 gametes. Dear students, now let's discuss the structure of gametes. First, spermatozoa or sperm. A sperm cell consists of 3 parts highly adapted for reaching and penetrating a secondary oocyte: a head, a midpiece, and a tail. The head is 4 to 5 micrometer long. It contains an acrosome, a lysosome-like vesicle, and a nucleus that has the haploid number of chromosomes. Enzymes within the acrosome include hyaluronidase and protease, which aid penetration of sperm cell into secondary oocyte. The middle piece has proximal and distal centrioles, the axial filament, and numerous mitochondria which provide energy for locomotion of the sperm. The tail is long and consists of the main piece and end piece, with central axial filament, fibrils, and partly covered by protoplasmic membrane, which helps to propel the sperm cell along its way. From head to tip of tail, human sperm are about 70 micrometers in length. Now let's discuss ovum. When the Graafian follicle is ripe, it projects from the ovarian surface, finally bursts and releases the mature ovum into the funnel of the oviduct. The mature ovum is roughly spherical, about 100 to 200 microns in diameter. The mature ovum has a nucleus and nucleolus, commonly called the germinal vesicle and germinal spot, respectively. The protoplasm of the ovum, called ooplasm, provides nutrition during early embryonic development. The cell membrane of the ovum is called vitelline membrane or oolemma. Since it appears as a colorless zone, it's also called the zona pellucida. The space between the vitellius or ooplasm and the vitelline membrane is called perivitelline space. When the mature ovum is discharged, some of the follicle cells are still attached radially around the ovum, called corona radiata. The ovum is propelled down the oviduct to the uterus by ciliated cells. This transfer takes several hours. Dear friends, now let's move on to the next topic, fertilization. Fertilization is a process whereby 2 sex cells or gametes fuse together to create a new individual with genetic potentials derived from both parents. Thus, the first function of the fertilization is to transmit genes from parents to offspring, and the second is to initiate those reactions in the egg cytoplasm that permit development to proceed. The male gamete is small cell with greatly reduced cytoplasm and haploid nucleus. Sperm usually has 2 distinct regions enclosed by plasma membrane, head and tail. The head contains nucleus and acrosomal vesicle. The DNA in the nucleus is highly condensed and transcriptionally inactive because the normal histones are replaced by the special class of packaging proteins known as protamines. The acrosomal vesicle is a specialized secretory vesicle present in most animal sperms and is derived from Golgi apparatus containing hydrolytic enzymes needed for digestion of extracellular coats surrounding the egg. The motile tail of a sperm is long flagellum. The motility of flagellum is caused by the sliding of microtubules driven by dynein ATPase activation. The ATP is generated by the large number of highly specialized mitochondria that are concentrated in the anterior part of the sperm tail, particularly in the midpiece. The female gametes of animals are larger than the sperm and contain materials needed for initial development of the embryo. The egg cytoplasm usually contains nutrient reservoir in yolk form, which is rich in proteins, lipids, polysaccharides, and yolk granules. Yolk synthesis takes place outside ovary and is imported to oocytes. The yolk concentration is large in birds, amphibians, and insects. The yolk proteins are made by liver cells, which secrete the protein into blood. Within the ovaries, oocytes use receptor-mediated endocytosis to take up the yolk protein from the extracellular fluid. Yolk is concentrated towards one pole of the egg, and the pole where yolk accumulates is called vegetal pole, and the opposite pole is called animal pole. The polar body is formed during oogenesis but of the animal pole. A placental mammalian egg is not as large as eggs of the birds. Because of this, the embryo starts growing early by taking up nutrients from mother via placenta. There is another peculiarity of mammalian egg, that is, it contains egg coat, which is a glycoprotein present immediately surrounding the egg plasma membrane known as zona pellucida. Although the details of fertilization vary from species to species, but it generally consists of 4 major events. First, a contact and recognition between sperm and egg. Second, regulation of sperm entry into the egg. Third, fertilization of the egg. Fourth, fusion of the genetic material of sperm and egg. And finally, activation of egg metabolism to start development. Mammalian sperms are initially not ready to accomplish this task. They need some modification to acquire the capacity to fertilize the egg. The modification takes place by conditioning in reproductive tract, and the process is known as capacitation. Capacitation alters the sperm in 2 ways. First, it greatly increases motility of the sperm. Second, it makes the sperm to undergo acrosomal reaction. During capacitation, the sperm undergoes functional and biochemical changes. It removes the cholesterol content. Besides, there occurs efflux of potassium ions, which results in a change in the membrane potential of the sperm, hence it becomes more negative. This change in membrane potential may allow calcium and bicarbonate channels to open and permit calcium and bicarbonate ions into the sperm, leading to shift in its membrane potential. Although sperm can fuse with membranes having a resting potential of -70 millivolts, they cannot fuse with membranes having a positive resting potential, so that no more sperm can fuse the egg. Dear students, now let's discuss them one by one. First, contact and recognition between the sperm and egg. The interaction of sperm and egg generally proceeds according to 5 basic steps. First, the chemoattraction of the sperm to the egg by soluble molecules secreted by the egg. In many species, sperm is attracted towards the egg by chemotaxis that vary in different species. However, in external fertilization, sperm motility is acquired when sperm is spawned in water bodies. Second, the exocytosis of acrosomal vesicle to release its enzymes. Third, the binding of the sperm to the extracellular envelope of the egg. Fourth, passing of the sperm through the extracellular envelope. Fifth, fusion of the egg and sperm cell plasma membranes. Recognition of the sperm by the vitelline envelope or zona pellucida is followed by the lysis of that portion of the envelope or zona pellucida in the region of the sperm head by the acrosomal enzymes. This lysis is followed by the fusion of sperm plasma membrane of the egg. Sperm to egg binding appears to cause the extension of several microvilli to form the fertilization cone. In the sea urchin, all regions of egg's plasma membrane are capable of fusing with sperm. In several other species, certain regions of the membrane are specialized for sperm recognition and fusion. Fusion is an active process often mediated by specific fusogenic proteins. It seems that binding plays a secondary role as a fusogenic protein. In mammals, the fertilizing proteins in the sperm plasma membrane are essential for sperm membrane to egg membrane fusion. The acrosomal reaction alters the sperm surface so that it can bind to and fuse with plasma membrane of the egg. After fusion, the contents of the sperm, that's nuclei, are drawn into the egg. Fusion with sperm activates the egg, leading to the cortical reaction. Upon fertilization, the intracellular calcium ion concentration of the egg increases greatly. In the high calcium environment, the cortical granules fuse with the egg plasma membrane, releasing their contents. The rise in the calcium concentration responsible for the cortical granule reaction is not due to an influx of calcium into the egg, but rather occurs due to the release of calcium from intracellular storage. The cortical reaction changes the structure of the zona pellucida so that sperm cannot penetrate in, and thus polyspermy is prevented. Polyspermy is a condition in which more than one sperm fuses with single egg. During the reaction, zona pellucida 3 is inactivated so that it can no longer bind to sperm and thus inhibits further acrosomal reaction. The second part, regulation of sperm entry into the egg. On encountering an egg, the sperm, which is capacitated sperm by now, must penetrate the egg's layer, that's zona pellucida. The egg has mainly three glycoproteins, which are zona pellucida 1, zona pellucida 2 and zona pellucida 3, all of which are produced exclusively by growing oocyte. The zona pellucida 3 induces sperm for acrosomal reaction in the mammals, while bindin acts as such in sea urchin. As soon as one sperm has entered the egg, the fusibility of the egg membrane, which was necessary to get the sperm inside the egg, is lost and prevents polyspermy. In sea urchins, as in most animals studied, any sperm that enters the egg can provide a haploid nucleus and a centriole to the egg. In normal monospermy, in which only one sperm enters the egg, a haploid sperm nucleus and a haploid egg nucleus combine to form the diploid nucleus of the fertilized egg, that's zygote, thus restoring the chromosomal number appropriate for the species. The centriole, which is provided by the sperm, will divide to form two poles of the mitotic spindle during cleavage. The entrance of the multiple sperm leads to the disastrous consequences in most organisms. The third part, fusion of the genetic material of the sperm and egg. In sea urchins, the sperm nucleus enters the egg perpendicular to the egg surface. After fusion of the sperm and egg plasma membranes, the sperm nucleus and centriole separate from the mitochondria and the flagellum. The mitochondria and the flagellum disintegrate. The haploid egg nucleus is called female pronucleus. The sperm nucleus inside the egg decondenses to form the male pronucleus. The sperm nucleus undergoes a dramatic transformation. The nuclear envelope vesiculates into small packets, thereby exposing the compact sperm chromatin to the egg cytoplasm. The proteins holding the sperm chromatin in its condensed inactive state are exchanged for other proteins derived from the egg cytoplasm. This exchange permits the decondensation of the sperm chromatin. In sea urchins, decondensation appears to be initiated by the phosphorylation of two sperm-specific histones that bind tightly to the DNA. This process begins when the sperm comes in contact with a glycoprotein in the egg jelly that elevates the level of CAMP-dependent protein kinase activity. These protein kinases phosphorylate several of the basic residues of the sperm-specific histones and thereby interfere with their binding to DNA. This loosening is thought to facilitate the replacement of the sperm-specific histones with other histones that have been stored in the oocyte cytoplasm. Once decondensed, the DNA can begin transcription and replication. In mammals, the process of pronuclear migration takes about 12 hours compared with less than 1 hour in the sea urchin. The mammalian sperm enters almost tangentially to the surface of the egg rather than approaching it perpendicularly, and it fuses with the numerous microvilli. The mammalian sperm nucleus also breaks down as its chromatin decondenses and is then reconstructed by coalescing vesicles. The DNA of the sperm nucleus is bound by the basic proteins called protamines, and these nuclear proteins are tightly compacted through disulfide bonds. In the egg cytoplasm, glutathione reduces these disulfide bonds and allows the uncoiling of the sperm chromatin. The mammalian male pronucleus enlarges while the oocyte nucleus completes its second meiotic division. The centrosome accompanying the male pronucleus produces its esters and contacts the female pronucleus. Then each pronucleus migrates towards the other, replicating its DNA as it travels. Upon meeting, the two nuclear envelopes break down. However, instead of producing a common zygote nucleus, as happens in sea urchin fertilization, the chromatin condenses into chromosomes that orient themselves on a common mitotic spindle. Thus, a true diploid nucleus in mammals is first seen not in the zygote but at two-cell stage. The fourth, activation of egg metabolism to start development. Although fertilization is often depicted as merely the means to merge two haploid nuclei, it has an equally important role in initiating the processes that begin development. These events happen in the cytoplasm and occur without the involvement of the nuclei. Contact of the sperm with the egg surface initiates metabolic reactions within the egg that trigger the onset of embryonic development. The chromosomes from the two gametes share a common spindle apparatus during the first mitotic division of the zygote. Only after the first division, as diploid nuclei form in the two daughter cells, the chromosomes from the two parents come together in a common nucleus. The zygote then divides into many smaller cells and the process is called cleavage. During this period, cells go through the S phase, that is DNA synthesis, and M phase, that's mitosis phase of the cell cycle, but may skip the G1 and G2 phases. The first 5 to 7 divisions form a cluster of cells known as morula. A fluid-filled cavity called blastocele forms within the morula, which becomes a hollow ball of cells called the blastula. The blastula undergoes gastrulation, which is the rearrangement of embryo into triploblastic gastrula, which is 3 germ layers. The embryonic germ layers are the ectoderm, which is the outer layer of the gastrula, the mesoderm, which fills the space between ectoderm and endoderm, and finally the endoderm, which lines the embryonic gut. Later, during organogenesis, the organs of the animal body form from the 3 embryonic germ layers that follows gastrulation and leads to the development of the new organism, and the cycle continues. Well, that's all we have in today's program. I really hope you would have understood. Thank you.